It's my great honor to introduce our work here. My talk will focus on functional effects of several human Mda receptor mutations. First, some background. It is well known that glutamate receptor mediate excitatory synaptic transmission in the brain. There are three types of aerotropic glutamate receptors, AMPA, kainate, and MDA, named after their selective agonist. Presynaptic stimulation of presynaptic neurons can generate this EPSC. The EPSC MDA component is a slow rising and long-lasting component which mediated mainly by the calcium ions. It is well established that MDA receptors are not only involved in the normal brain functions such as synaptic plasticity, learning, memory, and brain development, but also implicated in the wide range of pathological conditions, including epilepsy, Alzheimer's, Parkinson's, and schizophrenia. MDA receptors composed by two glutamate-1 subunit and two glutamate-2 subunit. There are only one gene product of glutamate-1 subunit with 8 splicing variants, but with 4 different types of the glutamate-2 subunit, 2A, 2B, 2C, and 2D. Activation of glutamate MDA receptors require both agonist binding. Once channel open, calcium and other ions can flow through. All the glutamate receptor, MDA receptor subunits share same architecture. We can this linear top view. The left part in green is the endothermic domain, and the blue part forms agonist binding domain. The yellow part is the transmembrane domain. The right part is the C-terminal domain. This panel is a crystal structure of tetrameric MDA receptors with side view oriented from the extracellular side to the intracellular side. The right panel is a cartoon for a single MDA subunit. We can see this agonist binding site looks like a clamshell. Agonist binding can use this closure of this clamshell cause movement. The movement can be transferred to transmembrane domain, leading to channel open. This is channel gating. Along with the progress of next sequencing technology in recent years, significant numbers of Mda receptor mutations were identified. This is our summary on all reported Mda receptors mutation which are not existing in NORMAD. Is a database for the healthy population. We can see the genetic variants occurs through all the MDA subunits with the 2B and the 2A most. The patient phenotype including ADHD, autism, epilepsy, intellectual disability, and schizophrenia, with the intellectual disability the most, followed by the epilepsy. The lower panel shows that these mutations are scattered across the whole gene from the endothermic domain to the C-terminal domain, with the agonist binding site and the transmembrane domain the most. Dr. Slavi Petrovsky, our collaborator from Australia, used this RVIS, residual variation intolerance score, to evaluate the genetic variation in the healthy population from the exact database. We can see that intolerant genes have fewer expected SNPs fewer observed SNPs than the expected ones. From this table, we can see most of the green gene is intolerant to the genetic variation. In the individual gene, we can see the different domains, so the different degree of intolerance. For example, this green 2A gene, we can see these 3 regions, pre-M1, M3, and pre-M4, so the least intolerance, reflected here by this smaller proportion of observed missense mutation compared to the expected one. Our work and other labs' work suggested these 3 regions, pre-M helix, M3 transmembrane domain, and also this pre-M4 domain are the control elements for the channel gating. Indeed, these small regions host significant numbers of disease-associated de novo mutations This is an incomplete list of published and unpublished missense mutation. Today I will focus on 4 missense mutations. The first one is this GRIN2A-L812M located in preM4. The patient has drug-resistant epilepsy from NIH. The patient also has a severe developmental delay. He also has continuous abnormal arm and leg movement. The brain MRI showed the cerebral atrophy and enlarged ventricles. The whole exome sequencing identified a missense mutation located between the agonist binding domain S2 and the transmembrane domain M4. This leucine residue is within van der Waals contact with pre-M1 helix and M3 cytolase motif. And the leucine to methionine mutation may interact with M3 element to change the channel gating. So as a step to understand the effects of this mutant on channel function, we evaluate agonist potency, current response, and single channel properties. Outside recording showed that this mutant has enhanced gluten potency and enhanced glycine potency by near 10-fold, which means the mutant receptor can be activated by much lower concentration of the agonist. Since the mutant of this patient is heterozygous and Mda receptor tetrameric has 2 copies of GRIN2A subunit, we anticipate that some receptor of this patient may contain a single copy of the mutant. We therefore employ a strategy by attaching this complementary set of coiled domain and followed by this ER retention signal to control the identity of the GRIN2A subunit on the cell surface. By this method, we can generate 3 combinations, wild-type 2A2A, the mutant 2A, and the mutant and the mutant. We repeated the experiment, which showed an intermediate but significant enhancement of both glutamate and glycine potency. This data indicate that a single copy of the mutant can significantly change the agonist potency. We also evaluate the effect of this mutant on sensitivity to the negative modulators. The data showed here, the mutant receptor showed reduced sensitivity to zinc inhibition, protein inhibition, and also to the magnesium inhibition. And also single copy of the mutant showed the dominant effect. We then performed whole cell Petroclap recording on transfected hex cell. We measured the time course by prolonged application or a brief application of the glutamate or glycine. Our data show that the mutant receptor showed a significantly slower deactivation time course by 8 to 10-fold to both glutamate and the glycine. We were curious about the effect of the single channel, the single copy of the mutant on the current response. We repeat the experiment on this trihatch L812M mutant. We can see the single copy of the mutant showed the intermediate to slow of the deactivation time course. We subsequently record the outside our single channel recording. We can see the single copy and the double copy both showed increased mean open time and increased open probability. Taken together, this mutant is a gain of function mutation. Indeed, the post-transfection of the mutant on the cultured hippocampal cells can induce the neurotoxicity, indicated here by these blabs. Since the classical anti-epileptic drugs did not provide adequate control of patients' seizures, we evaluate a set of FDA-approved Mva channel blockers, about 35 drugs. The drug highlighted here showed comparable sensitivity at the mutant receptor compared to the wild type. Among them, memantine was chosen because it's a safety in the pediatric patient. It's an anti-convulsant effect on almost all the animal model of epilepsy, and also its ability to inhibit the mutant receptors. adding on memantine can significantly decrease the seizure activity with the EEG improvement. In addition, the patient's abnormal continuous arm and leg movement were stopped completely. The next two mutants I want to introduce are located in this pre-M1 helix. The protein at the same position of GRIN2A and GRIN1 were mutated to arginine, can have 2AP552R and GLUN1P557R. The GRIN2AP552R was from a patient with epilepsy and developmental delay, and the GLUN1P557R is from the patient with severe intellectual disability with no seizure detected. You can see the location. This located in this 7-amino acid 2-run helix we call pre-M1. The protein at this position is all conserved through all the MDA subunit. This protein to arginine mutation may interact with this M3 transmembrane domain to change the channel gating. Our outside data showed that the mutant receptor has 10-fold and 20-fold enhanced glutamate and glycine potency, and a single copy of the mutant showed intermediate but significant enhancement of the glutamate and glycine potency. We then did whole cell patch lab on the transfected hex cell. You can see the mutant receptor showed slower deactivation time course to both glutamate and glycine. Surprisingly, the mutant receptor has 60-fold slower response rise time. This data suggests that this mutant can influence the channel activation rate and agonist dissociation. We are curious about a single copy of the mutant on this response time course. Therefore, we repeated the experiment on the trihedral P552R receptors. Our data show that a single copy of the mutant has a rapid rise time, although it has a slower deactivation time course. The double copy of the mutant has a slower rise time and also slower deactivation time course. This data indicate non-equivalent contribution to the gating by the 2GLUN2A pre-M1 helix. Similar to the P552R, the GLUN1P557R showed enhanced glutamate and enhanced glycine potency, also has a slower deactivation time course, but with the rapid rise time. This data indicate that the perturbation of GLUN1 pre-M1 helix do not change the time course of the channel activation. So we subsequently compare the current amplitude normalized to cell capacitance. This data is from transfected hex cell. We can see 2AP552R has comparable current amplitude to the wild type, but GLUN1P552R has a significantly smaller current amplitude, one co-expressed with 2A and co-expressed with GRIN2B. We then did surface protein biotination and subsequent Western blot to confirm if the reduced current amplitude is caused by the receptor trafficking. We can see the one co-expressed with GRIN2A, the P552R showed decreased ratio of the surface to total protein level. You can see it here by this bar graph. While the 2A P552R showed comparable surface expression compared with the wild type. One co-expressed with the GRIN2B, the P552R, the total protein level of the P557R significantly decreased, which is consistent to the reduced current amplitude. This data suggests that the GRIN1 P557R is maybe a loss function mutation and may underlie certain features of patient's phenotype of severe intellectual disability. Since the 2AP55R showed highly unusual response time course, therefore we sent it out for further investigation. We did outside of the single channel PET clap on trihydra P552R receptors. You can see single copy of the mutant showed no significant change in immune open time and the channel conductance level, but the double copy of the mutant showed increased immune open time with the decreased the channel conductance level. The increased charge transfer by this P552R mutant raises the possibility that this mutant may be neurotoxic. We therefore, collaborating with Dr. Isman lab at Pittsburgh University, we performed the neurotoxicity experiment on cultured cortical neurons. We can see co-transfection of the P552R can produce rapid and significant swelling of the dendrite, indicated by these numerous blebs. Inclusion of memantine in the cultured medium can reduce this dendrite swelling and also can rectify the decreased viability. This data suggests that memantine can protect the cultured neurons from P552R-induced neurotoxicity. Indeed, our rescue pharmacology experiment by oocyte recording showed that the FDA-approved channel blockers has a similar or even better potency or sensitivity for the mutant receptor compared to the wild-type receptors. We made the transgenic mice on this mutant. The homozygous mice has spontaneous seizures and died from the lethal seizures usually around P20 to P25. The heterozygous mom does not take care of the pups, and the heterozygous dad seems more aggressive. That's make our study progress very slow. We did have chance to do whole cell PET lab recording on hippocampal CO1 cell. By stimulating the Schaeffer collaterals, we recorded this evoked synaptic events. We can see the heterozygous neurons has a 5-fold larger leak current, which is APV sensitive. If we compare to the wild type, we can see the heterozygous neurons has 5 times slower deactivation decay time course and 4 times slower of the rise time. This data is pretty much preliminary. The last mutant I want to introduce, this is S2AS644G, located in the M3 transmembrane domain. The patient mom has abnormal pregnant history, and the patient has epilepsy and also has a severe developmental delay. The mutation serine to glycine occur in this M3 site 11 motif, which is a very important element to control the channel gating. Again, our oocyte recording data showed that the mutant has more than 10-fold enhanced of glutamate and glycine potency. Also almost abolished the zinc inhibition and the protein inhibition, but no change on the magnesium inhibition. The PET lab data on the transfected hex shell showed more than 10-fold slower of the deactivation time course, and also has a significantly increased open probability. Again, this is a gain of function mutation. Our collaborator, Dr. Franco, at Columbia University made this S644G transgenic mice. You can see the transgenic mice, the homozygous mice died of spontaneous lethal seizures with 15 days of mean survival. And a standard electroconvulsant threshold test showed that this heterozygous mice has reduced the threshold to this maximal tonic hydrogen extension seizures. And HE staining showed the reduced thickness of the hippocampus in both homozygous and heterozygous mice. And also this multi-electrode recordings on the cultural cortical neurons showed prolonged duration of the electrically evoked burst for both homozygous and heterozygous mice. Okay. Summary. GRIN mutations are involved in the subset of patients with epilepsy. There is a mismatch between the high volume of genetic data and functional evaluation. Transmembrane domain mutations can increase Mda receptor function in multiple ways, leading to Mda receptor hyperactivity. That's a channel opens more often, and receptors activated by lower concentration of the agonist, and also synaptic response endure longer, and the channels are less sensitive to negative modulators. Functional evaluation mutations can provide individualized strategy with potential anticonvulsant and neuroprotective actions. And last, disease-associated human Mda receptors identified the channel gating elements and advance our understanding of the receptor function. So here, first I'd like to thank Dr. Stephen Chandler, was my mentor and also now is my close collaborator, encouraged me apply grants to be independent. And Sharon is our postdoc fellow, did the neurotoxicity experiment and also some part of the Western blot. Chad is talented PhD student, did P552R transgenic mice slice work. And Wendy Chen is visiting MD PhD student from China, did a lot of oocyte recording, wholesale single channel recording, and also did some Western blot. And currently we are collaborating with 40 medical centers on this human Mda receptor mutations. The first mutation, L812, is from Williams School at NIH. The S644G mutant is from Emory from Harvard. And Dr. Juan Franco and David Goldenstein groups made transgenic mice and also did a functional study on S644G mice. And Dr. Ismail lab at Pittsburgh helped on the neurotoxicity experiment. And computational chemist at Emory, Petersburg, helped us on the modeling. And my work supported by NIH and Emory and Atlanta Children's Hospital. And currently I'm supported by Sage Company, studying on neurosteroid on Mda receptor, which has no relation with my talk. Thank you very much.
Thank you, Hongjie. Questions? Yeah.
So the PP552R, it has an increase over probability like all the other mutations, but also has a smaller conductance.
Yeah.
How do you explain the smaller conductance?
Yeah, I think probably because that is proline to arginine may change the side chain volume, may change the M3 position. The second possibility, probably this change the residual charge caused this change.
So is the calcium permeability affected in that mutation?
We did not check that.
Derek.
Yeah, very nice talk. Because NMDA receptors are so important in neurodevelopment, we think that most of these mutations that you've been studying with Steve over all the years, they really set up, it's the developing circuit that's messed up. And so all the clinical features that you eventually get are because of what happened way back during development. Have you or Steve tried to look at that in any way?
Yeah, that's why we kind of now, to make a series of transgenic mice. This two is a gain of function. We are making 2 or 3 loss function. We want to see, how at circular level, how the mutant influence the circular, because most of the data is from the in vitro. So.
Yeah. Have you tried to do conditional knockout mice so that you knock out or you introduce the mutation later on after development?
We don't do that, but I think it's a good idea too.
I mean, I guess for the P552 that you introduced, the homozygote is dying at P14. That's exactly the time you'd expect 2A to be coming up.
Right.
So my question is, earlier in development, if you looked at a sort of first postnatal week, is there actually any phenotype there, electrophysiological phenotype in these where 2B dominates? Do you see a difference at that earlier stage when there's relatively little 2A?
No, I think we are making 2 or 3 2B transgenic mice, see what happens. And also interestingly, loss function of GRIN2A mutation is also related to epilepsy. So we are thinking about probably because the 2A, 2B, there's a switch. Early stage is 2B. With a grown up, 2A coming and 2B going down. If 2A is a loss function, maybe that switch will not happen. 2B is still keeping high level. That may contribute hyperactivity.
There was a question here, yes. So.
Do the different mutants differ in their pharmacology?
Yes. Yeah, I think most of them, yes. That's why we need screening, set of FDA compound. Most of them, yes.
Ben.
Do any of the gain of function mutations in 2A that you've looked at, if you do the same ER retention and forced triheteromeric receptors, are they obvious in a 2A, 2B situation?
Yeah, that's a good question, but we only, in the vitro experiment, we only evaluate within 2A. 2A wild type, 2A mutant. But the reality probably is, the mutant 2A with wild type 2B. I think that's a very good question, yeah.
Okay, last question.
Do you know how this mutation affects synaptic plasticity, long-term fixation, long-term depression in the slice preparation?
We don't do that, but I think it's a good suggestion, yeah.
Martin, I saw you had one.
Yeah. So memantines are fairly well tolerated drug, so there's good precedence here from data to do some investigator-led trials.
Yes.
So individual trials in these patients. So can you give a greater flavour for how effective that's been in some of this early work?
I think at least till now we have 3 cases. It has effect at certain degree to control patients' seizures, but we also have at least 2 or 3 has no any effect on. That's why we currently has a cure grant to do a clinical trial. We want to see the profile, how memantine, ketamine, and other FDA drugs, working on this different type of human mutations.
Okay.
Thank you very much.
Thank you very much, Hongjie.